Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello, Senior 4 students. Welcome to TV Learning Program. My name is Benon. I will be with Senior 4 students in Biology. Dear students, remember our subject is still about life because biology is the study of life. And when we talk about life, we mean life of living organisms. Living organisms have their structures, and those different structures perform different functions which make organisms survive. Now, this lesson of today is from Unit 10, and Unit 10 is about enzymes, and this lesson is about naming enzymes. Then, students, by the end of this lesson, make sure you are able to define the term enzyme, explain the functioning mechanism of enzymes, and describe factors which affect the rate of enzyme activity. Dear students, what do you understand when we talk about the term enzyme? Enzyme is a protein catalyst which speeds up the rate of chemical reaction in the body. Meaning, enzymes are proteins in nature, and they speed up chemical reactions in our body. Let us have the common understanding about enzymes. Enzymes work on what we call substrates, and these substrates are converted into products. So, which means enzymes do not change, but the substrates are converted into products, which means after the reaction, the enzyme will not change, but the substrate will change into a product. A substrate is a molecule upon which the enzyme acts on. In case of a single substrate, the substrate binds with enzyme active site to form what we call enzyme-substrate complex. Thereafter, the substrate is transformed into one or more products, which are then released from the active site of the enzyme. Let us take an example here. This one is enzyme plus substrate. Then they form enzyme-substrate complex. Remember, the substrate will fit into the active site of the enzyme. Then from there, this enzyme will catalyze the substrate. Then we will have enzyme product formed, from where the enzyme and product will be separated. That means after the reaction, we get the enzyme, and then substrates have already been converted into products. Enzymes are protein catalysts or biological catalysts which speed up the rate of chemical reactions in the body. Let us take examples of enzymes. We have defined the term enzyme, and these enzymes are very many in our body. Let us see some of them, those which are common in our body. We have like enzyme amylase. We can have another example of maltase. We can have enzyme lactase. We can have enzyme sucrase. Then we have enzyme pepsin. We can have trypsin. And we can have DNA polymerase, and so on. These are examples of enzymes which are commonly found in our body, and they perform different functions. Let us take an example. The function of amylase is to break down starch to maltose. Remember, starch, we get it from energy-giving foods like carbohydrates. So this starch is broken down into maltose by enzyme amylase. Then we also have maltase enzyme. The function of this maltase is to break down maltose. Maltose is formed from starch, and then that maltose is broken down by maltase enzyme into two glucose molecules. Then remember, glucose is simple. It can be absorbed by the body so that the body will use it as the source of energy. Then we also have lactase enzyme. This one breaks down lactose into glucose and galactose. Those are simple sugars, or we can even call them monosaccharide sugars, which can easily be absorbed by our body. Then we have sucrase enzyme. The function of this enzyme is to break down sucrose, which is a disaccharide sugar, into glucose and fructose. Then glucose and fructose are simple sugars which can be absorbed by our body. We have pepsin. This one breaks down proteins into amino acids. Then we also have trypsin, the same. It breaks down proteins into amino acids. Then we have DNA polymerase. This one joins strands of DNA to form a large strand. When you see, most of these enzymes end with ASE. So one of the common characteristics or ways we can easily identify these enzymes is to check if their names end with ASE. But remember, not all enzymes end with ASE. Now that we have seen what happens when enzyme combines with the substrate to form enzyme and product, let us see different views of scientists how enzymes work in our body. One of them is lock and key hypothesis. This is a hypothesis. In this hypothesis, the substrate is considered as the key, and the enzyme is considered as the lock. Dear students, remember in your homes you have what you call locker, and then you have the key which opens the locker. So in this case, we say the key you use to open is the substrate, and then the enzyme is the lock or the locker. So enzymes have what we call active sites, and the active site is exactly complementary to the shape of the substrate. Let us see it here. As you see, this is the enzyme, and this enzyme has what we call active site, and in that active site, that's where this substrate is going to enter. You can see that the shape of the substrate will exactly fit into the active site of the enzyme. When the substrate binds into the enzyme, they form what we call enzyme substrate complex. Like you see, this is the enzyme, and then this is the substrate, and they are binding together. What happens? The enzyme changes the shape slightly so that the substrate can fit there. So when the substrate fits there, there is formation of enzyme substrate complex. From there, the enzyme is going to catalyze the substrate. What will happen is enzyme product complex. Like you see, the substrate has been catalyzed, and then the products here, we are going to see the enzyme we started with, and then the products which have been formed from the substrate. Then products are going to leave the active site of the enzyme so that this enzyme is able to catalyze another substrate. So this process where the substrate fits into the active site of the enzyme, we call it lock and key hypothesis, because you know how the key fits into the locker so that you are able to open that locker. Remember, if you bring another key, it will not be able to open that locker. So there is a specific key for a specific locker. So this is the same way enzymes work in our body. So we call it lock and key hypothesis. Then we have another one, which we call induced fit model. To induce means to convince, to persuade, or to get. So in this hypothesis, the active site of the enzyme, remember the active site we talked about, is flexible and not fully complementary with the shape of the substrate, which means the active site is not complementary to the substrate. An enzyme will collide with the substrate molecule and binds to the active site. This induces, this convinces, this changes the shape of the substrate so that it can fit into the active site of the enzyme. So a slight change in the shape of the enzyme, making the substrate fit more precisely into the active site of the enzyme. The products formed move away from the active site, and the active site of the enzyme regains its original shape or configuration, ready to be used for the next reaction which can take place. Let us see the induced fit model. When you see the shape of the active site is not complementary to the shape of the substrate. So what happens? The enzyme changes the shape slightly so that the substrate binds there. Then when the substrate binds there, it is catalyzed by the enzyme so that we get enzyme products complex. Like you see, these are the products, and then this is the enzyme. From there, the products are going to leave the active site of the enzyme, and then the enzyme will be ready to catalyze another substrate. So from there, we have two models. We have what we call lock and key hypothesis, which we talked about. Then we also have induced fit model. So that in the induced fit model, the active site of the enzyme is not complementary to the shape of the substrates. But what happens? The enzyme changes the shape slightly so that the substrate can fit into the active site of the enzyme. Now, students, we have seen examples of enzymes. We have seen different models describing the physiology or the functioning mechanism of the enzymes. Let us talk about factors which affect the rate of enzyme activity. Enzyme activity can be affected by a number of factors. One of them is temperature. These are the factors which affect enzymes. The factors which affect the rate of enzyme activity. We have one of them is temperature. We also have pH. We have enzyme concentration. We also have substrate concentration. Then we can have inhibitors. These are the factors which affect the rate of enzyme activity: temperature, pH, enzyme concentration, substrate concentration, and inhibitors. Let us see how these factors affect the rate of enzyme activity. Here we mean the factor may increase the functioning of the enzyme, it may decrease the functioning of the enzyme, or it can even make the enzyme not work at all. So this is what we are going to see. Let us start with the temperature as the factor which affects the rate of enzyme activity. Enzymes have what we call an optimal temperature at which they work. That means at optimum temperature, enzymes catalyze the substrates well. It's optimal because it is when the substrate matches exactly with the shape of the active site. That means the substrate fits well in the active site of the enzyme. Then, if the temperature is too high, that means beyond the optimum temperature, the enzyme will be denatured. What will happen? The active site of the enzyme will change the shape so that the enzyme will not be able to catalyze the substrate because the substrate will not fit into the active site. Now, this is our graph. On y-axis, we have increasing enzyme activity, and then on x-axis, we have increase in temperature. This temperature is in degrees Celsius. Then, as you see, as the temperature increases, the rate of enzyme activity also increases. At around 40 degrees Celsius, we have what we call optimum temperature, where enzymes' active site are able to receive the substrate, and then the rate of reaction will be high. Going beyond 40 degrees Celsius, that means enzymes will start to be denatured. Their active sites will change the shape, and substrates will no longer be able to fit into the active site. And what will happen? Enzymes will be denatured, and they will not be able to speed the rate of chemical reaction or to catalyze the substrates. So temperature is a factor which affects the rate of enzyme activity. Actually, they say optimum temperature for most of the body enzymes is around 37 degrees Celsius. Then we have pH, which is the potential of hydrogen. Remember, pH is measured from 1, then we have 7, up to 14. 7 is the neutral point. And from 7 going to 1, that means acidity increases. Then from 7 going up to 14, alkalinity or basicity will increase. So in that case, we have some enzymes which work well in acidic medium and others which work well in alkaline medium. So here they say enzymes also have an optimum pH level. So the large deviation in pH will cause the enzyme to denature. That means the change in pH will also denature enzymes and they will not be able to catalyze the substrate. For example, if the pH of blood changes from its average of 7.3 to 7.4 pH. That means the average pH of blood is blood pH, our blood, is between 7.3 to 7.4. So the large deviation, the change from this pH will affect the physiology of the body. And actually, if it changes too much, from 7.3 going down or from 7.4 going far above, that will change the physiology of our body, and it is possible, or it can lead to death. So here we have the graph. We have pH from 1 up to 7, then from 7 up to 14. So as the pH increases, the rate of reaction will also increase. And at around 7, we have optimum pH, where most of enzymes work nicely. At that level, enzymes are able to catalyze the substrates very fast. Going beyond, we'll start to denature the enzyme. That means the shape of the active site of the enzyme will change, and the substrate is no longer able to fit into the active site. So what will happen? Enzymes will be denatured, and they will not catalyze the substrate. So pH is a very important factor. Another one is substrate concentration. Let us say we have enzymes in our body. So when we increase substrates, that means when there are more substrates available, the reaction rate will continue to increase until all the active sites are occupied by the substrates. When at this point the reaction rate becomes constant, meaning we have substrate concentration. As the concentration increases, the rate of reaction will increase, and when all the active site of the enzyme is occupied by the substrate, the rate of reaction will be constant because active sites will be saturated with substrates, meaning substrate concentration is another factor which affects the rate of enzyme concentration. Then we have another factor, which is enzyme concentration. When we have substrates, as we increase the concentration of the enzyme, we'll speed up the rate of reaction as long as there is the substrate available to bind to the active site of the enzyme. Once all of the substrate is bound, the reaction will no longer speed up. It will be constant. So this is the graph describing the rate of reaction, that as the enzyme concentration increases, then the rate of reaction will also increase up to the time when all substrates are bound to the active site of the enzyme. Then we have inhibitors. Enzyme inhibitors are substances which alter, which change the catalytic action of the enzyme and consequently slow down, or in some cases stop catalysis. There are three types of common enzyme inhibition. We have what we call competitive inhibition, we have what we call non-competitive inhibition, and then we have substrate inhibition. Let us see these forms of inhibitors. We have competitive inhibition. Dear student, this is our enzyme. Then this is the substrate. Then this is the inhibitor. You can see the inhibitor and substrate can compete to enter into the active site of the enzyme. So when the inhibitor enters into the active site, that means the substrate will not be able to come and enter into the active site. So what will happen? There will be no enzyme substrate reaction because the inhibitor has entered into the active site. Another one is non-competitive inhibition. That's where we have the inhibitor, and then we have the enzyme, and then the substrate. What happens? The inhibitor doesn't go to the active site of the enzyme. Instead, it goes to another part of the enzyme, which we call allosteric site. And what will happen when the inhibitor enters there, it changes the shape of the active site of the enzyme, and then the substrate will no longer or will not be able to enter into the active site. Here it means the inhibitor doesn't compete with the substrate to enter into the active site. So in that case, the inhibitor will not allow the substrate to bind into the active site of the enzyme. Dear students, let us make a quick review of what we have talked about. We have seen what enzyme is. We have seen different hypotheses about the functioning mechanism of enzymes. Remember, we have seen lock and key hypothesis. We have also talked about induced fit model. Then from there, we have talked about 5 factors which affect the rate of enzyme activity. We have talked about temperature, pH, enzyme concentration, substrate concentration, and inhibitors. Dear students, this is the homework to test your level of understanding. If you followed nicely all the steps of the lesson, this is the homework. Question number 1, what is the meaning of the following terms related to enzyme activity? Catalyst, lock and key hypothesis, activation energy, and Q10. Question number 2, why are there hundreds of different enzymes in a cell? Remember, in our body we have very many enzymes. Why do we have them, and why are they important? Question number 3, how do enzymes reduce the activation energy of a reaction? Then we have question number 4. This is about the graph. You will plot the data on a graph, and then you will have to identify the optimum temperature of the reaction. Then you will describe the shape of the graph between 10 and 40 degrees Celsius. Then you will calculate the rate of increase between 20 and 30 degrees Celsius. Dear students, thank you very much for your active participation. Stay safe and enjoy the rest of the day. Bye.